Hello, everybody. My name is Daniel Pando. I am the CEO and one of the co-founders of Nanovex Biotechnologies and its cosmetic division INdermal. We are here today to talk about exosomes in cosmetic applications. Probably most of you already knows our company, Nanovex. Nanovex is a company that we founded 9 years ago to develop drug delivery systems for medical applications. And 3-4 years later, after creating the company, we decided to create INdermal, our cosmetic division, where we take this technology from medical applications and we adapt it to cosmetic applications in order to develop new delivery systems which they are able to increase the effectiveness of the encapsulated actives. So as I told you before, the effectiveness of the encapsulated actives with our technology can be increased by several times, 20 or the latest product could increase even 40 times. Then the protection of the encapsulated active is very important too. For example, in our new product, the Vegan DDS Pure Retinal, we have checked how when the retinal is encapsulated, the stability test at forces conditions showed that the product is protected, the molecule is intact after 4 weeks at 40 degrees. The integrity of the molecule is 95%. However, when the retinal is free, when the molecule is free, the integrity of this molecule in the same conditions, 4 weeks at 40 degrees, is less than 10%. This gives you an idea of the level of protection that our systems can offer. Then you can also mask some organoleptic problems related with color and other. And all these benefits are because we have different delivery systems based on liposomes and innovative liposomes, which we develop specifically thinking about the target where these actives have to be released, and also thinking about the encapsulated active. In the case of, you can see here, the main systems, the vegan deep delivery system is the system to release actives in the epidermis. Then we have the current delivery system to release actives on the surface of the skin, in the follicle, and finally in the hair. These are the main systems, but then we have other systems with higher specificity. For example, we have systems which could release the actives in the sebaceous gland or in the scalp or in the dermis, depending on the active, okay, and the properties that the customer is looking for. Of course, our technology is 100% vegan friendly, but today we are here to talk about exosomes. And exosomes are not new for us, okay, because from the beginning, several of our developed delivery systems have been based on these biomimetic exosomes. In fact, their importance is that the 2 letters of our name, the 2 letters at the end of Nanovex, this EX is because the exosomes, and we have been using this system during these 9 years to develop different therapies of delivery systems for the treatment of different diseases, including skin diseases. But what is an exosome? Exosomes are natural nanovesicles secreted by cells, okay, and for their communication. And they consist in the lipid bilayer enclosing an aqueous core that contains biocompounds like nucleotides, proteins, and other molecules. You can see also, check here, the difference between a liposome and an exosome. As you can see, which is the shell, the structure of the vesicle is the same. There are lipid bilayers formed by phospholipids, lipids, other compounds. This part is the same in the exosomes and the main difference is in the actives that the exosome contains, such as these proteins that could be in the membrane or inside, as well as the rest of the compounds I told you before, the RNA, other metabolites, among others. The history about the exosomes starting in 1967, Halbering et al. Described exocytosis of nanovesicles formed in multivesicular bodies into the cells. Okay, it was 16 years later when it was reported the first time small vesicles around 50 nanometers in size secreted into the extracellular space. And this time they start to think that these vesicles they are considered somewhat important. But finally, in 29, I mean that almost 50 years later, in comparison with 1967, it was the first time that was reported the plant-derived exosomes isolation. So this gives us an idea of the complexity of the isolation and the purification of these exosomes. And as you may have correctly guessed for the previous slide, exosomes are not an exclusive mechanism of animal cells. They are produced by many organisms, such as animals, plants, and microbes. Exosomes from plants, or how they are usually called, plant-derived exosomes like nanoparticles or nanovesicles, are quite important in cosmetics, and we will see why. Okay, this is the main structure composition of exosome, and exosomes are heterogeneous vesicles. Their composition depends on the status of the cell and the tissue of origin. For example, some proteins depend on the tissue and the status of the cell, but some others are non-specific. Plant exosomes are usually up to 500 nanometers, but normally they are smaller, around 300, 200 nanometers in size, and they are negatively charged to neutral. As mentioned before, they are basically lipid bilayers which contains phospholipids in the membrane, in the lipid bilayer. And in this case, exosomes also contain not too much, a few membrane proteins, mainly aquaporins and chloride channels. Okay, and finally, plant exosomes are rich in genetic material. However, it is estimated that up to 75% of this genetic material is unknown. Let's explore what's the main difference between the animal exosomes and plant-derived exosomes. And there are, in addition to this difference in the proteins in the membrane, which in the case of the animal exosomes, they contain higher amounts of transmembrane proteins. In the case of the plant-derived exosomes, these exosomes contain less or lower amounts. While animal-derived exosomes contain growth factors and cytokines, in the case of the plant-derived exosomes, they contain small molecules compounds with therapeutic effects. This is very important. Then, probably plant-derived exosomes are more stable to temperature and they have a lower immunogenicity. Okay. Other important difference is that the animal exosomes contains cholesterol as membrane stabilizer. In the case of the plant-derived exosomes, of course, they do not contain the cholesterol because they are vegan friendly. Okay, and what's their role in cosmetics? Well, they are key communicators in nature. Okay, as we have seen, not only are they able to communicate between cells of the same species, but some kingdoms. I mean that can work as key modulators in the gene expression of not only in plants but also in human cells. Here in INdermal, we have developed the technology to deliver the nanovesicles or the liposomes into different targets. As you can see here, we are able to release actives in the epidermis, on the surface of the skin, the follicle, and in the hair, as I told you before. So combining the natural properties together with this technology, we can produce very great exosomes like or biomimetic exosomes with ability to release their cargo, these biocompounds, genetic material, and between other compounds into the specific target. And also it's important to say that the plant-derived exosomes can enter the cells and deliver its content intracellularly. There are different ways how the exosomes can penetrate into the cells because they can use endocytosis, pinocytosis, phagocytosis, or because the similar composition in the membrane of an exosome, the same for a liposome, the similar composition of this lipid bilayer regarding the composition of the membrane of the cells, they can also use the fusion strategy, which the membrane of the exosomes and liposomes are fused with the cell membrane, allowing the release in the cytoplasms or of the encapsulated actives. So, as I told you before, these microRNA packaged into the plant-derived exosome vesicles can be the key modulators of human gene expression, and this is linked to a lot of different types of properties for human health and more concretely for cosmetic or for the skin interest. For example, there are several scientific articles which report anti-aging, good healing, anti-inflammatory, antioxidant, or antibacterial properties. Here you can see in these slides how is the mechanism, how these plant-derived exosomes are reported that they have these properties, in this case, the wound healing properties, for example, and you can see how the most of the properties are related with the increase of some proteins or the reduction of others. I mean that it's proven that they can work as key modulators of gene expression, allowing the production of some compounds or reducing the expression and the production of others. In the case of the wound healing properties, although these properties also work for other purposes, they can increase the production of collagen, the production of hyaluronate synthase in fibroblasts. They induce the proliferation, different types of properties. In the case of the anti-aging properties, also reported in several scientific articles, it's because they can, for example, decrease the expression of the gene associated with the metalloproteases, which are they are in charge of cause discomposition of collagen and skin aging, but however, they also can increase the expression of other genes, such as the FGF12. Other properties, as antibacterial properties, has been reported also in several scientific articles. In the case of the anti-inflammatory properties, there are also a lot of different studies which showed how, for example, in the case of the ginger, 27 microRNAs that are highly expressing and targeting many cytokines genes associated with inflammation. Okay, so finally they can regulate these genes associated with inflammation. Okay, also a lot of different antioxidant properties, but finally, in brief, what we want to show here is that plant-derived exosomes can work as key modulators of gene expression. Okay, so they, of course, they can be very, very interesting for the skin health. And now, okay, we have been talking about the different the composition of the exosomes, their properties, but how we can obtain these exosomes. There are mainly two different ways approach to obtain the exosomes. The top-down approach, which are this approach is to obtain directly the exosomes from the plants and then carry out different types of purification and isolation processes, such as forces to pass through membrane pores or microfluidic devices. Then carry out different types of separation, such as differential centrifugation, or also further purification using density gradient ultracentrifugation. Okay, so finally, the goal here is to obtain the exosomes directly from the plants and trying to purify and to isolate the exosomes. It is very difficult because exosomes in plants are not in plants, in the most of the organisms are in a very, very, very low concentration. Okay, very, very low concentration. So it's very difficult to have exosomes at high concentration to be used in cosmetic applications. We will see later that this will be one of the main problems of this approach. The obtention of the exosomes directly from plants involves that or is linked to a products with very, very low concentration of exosomes. However, There is other alternative. There is the bottom-up approach in which the plant-derived exosomes are generated fully in the lab using supramolecular chemistry. In this case, we obtain from the plants, I mean, this is the way that we use here at INdermal, we obtain from the plants all the compounds that then will form an exosome in this case. It is linked with a lot of different benefits that we will see in this slide, in which we compare the advantage and disadvantage of the plant exosomes. Okay, as I told you before, the main problems are the very low concentration, and of course, there is no possibilities of customization, because when you when you obtain the exosomes from the plants, they are already formed, so you cannot encapsulate actives inside. You could carry out some kind of slight modification on the surface of the exosome by adding some proteins using bioengineering tools, but you need a very, very high grade of purification and isolation of the exosomes, and apply these tools, these bioengineering tools, is so expensive that it makes no sense at all. Other way is to obtain the biomimetic exosome using this bottom-up approach, as I told you before. So the main benefits, or this way contains the same benefits that the other plant exosomes, because as we are obtaining all the raw materials to form the exosomes directly from plants, our exosomes are also 100% natural and 100% obtained from plants. But in this case, we have a very much higher concentration of exosomes. Concretely, we can develop products with 1 million more, a concentration 1 million times more higher, sorry, than the exosomes from plants. This is a very huge difference to have in a milliliter 1 million times more concentration of exosomes. Okay, and also we have the possibility of make a total customization of the exosome. I mean that we can, as we are producing the exosomes here, we can encapsulate different compounds inside. I mean that we can combine the properties of different natural plant exosomes, encapsulating in the same exosome different biocompounds from different plants, or even other type of molecules. So, in our opinion, okay, the main limits or limitations of plant exosomes are the very low exosome concentration, around 10^8 power exosomes per milliliter. There is a kind of waste of potential, because the exosomes are excellent vectors for transportive actives. However, as in this case, the most of the space of the exosomes are not used. I mean that there is room enough to encapsulate more actives, but you can't, because the exosomes that you are already obtaining, the exosomes that you are obtaining, they are already formed, so you cannot encapsulate, you cannot customize, and then this production of the plant-derived exosomes are often affected by natural factors such as the climate, the season, the region, and also the status of the plant. However, this biomimetic-like system, okay, in fact, of this very much higher concentration, 1 million times more exosome concentration, up to 10^14 power exosomes per milliliter, you can also vectorize in the same exosome different types of compounds. There is no limits here, because you can request or you can ask us about to encapsulate the biocompounds from this plant to the biocompounds of these other plants, or even other type of compounds which could have synergistic effects with these biocompounds. Finally, you can customize, you can carry out a full customization of the exosomes, and related with the manufacturing, there is a more homogeneous exosome production, and there is no variations between batch to batch. We are now developing 3 different products. Okay, these products will be launched in March of 2024, hopefully. The first product will combine. It will be a product for hair growth, which combines the biocompounds from the Trifolium pratense, the red clover, which it is well known that this plant, these biocompounds from these plants contribute to reduce the alopecia. Okay, and we are also include in this exosome the acetyl tetrapeptide 3, which stimulates the extracellular matrix proteins, and this is key for the maintenance of good hair anchorage and also to reduce the inflammation. The second product will be an anti-acne product, which combines 4 different biocompounds or 4 different plants, the biocompounds of these 4 different plants, obtaining finally a lot of different properties with very high interest for the treatment of the acne, such as antibacterial, anti-inflammatory, antioxidant, anti-acne, anti-irritant, hydration, different types. And we are combining all these biocompounds from this plant with these benefits with azelaic acid, natural origin azelaic acid, which has also very interesting properties for acne, such as the anti-inflammatory, antioxidant, and antibacterial, both for gram-negative and gram-positive. And finally, we are developing a product for anti-aging with anti-aging properties, which contains biocompounds from Pinus pinaster with antioxidant, anti-inflammatory, and antimicrobial properties, with, in this case, bakuchiol, which is known to be like the natural retinol. Okay, I mean that it works, or it has similar benefits in comparison with the retinol, mainly the stimulation of the collagen and elastin synthesis. But probably the most important part here is the ability to customize the biomimetic exosome-like products. You know that at INdermal, we are experts in carry out different custom products based on liposomes, exosomes for our customers. And in this case, there are even more possibilities of customization in comparison with the liposomes, because in this case, we have more actives. We have of course, the same than the liposomes. We can modify the structure of the exosome to develop an exosome that could release actives in the epidermis or on the surface of the skin or in the follicle or in the sebaceous gland, depending on the requirements of the customer. But then, okay, we can also customize the proteins that are included into the exosome, in general, the biocompounds of the exosomes, and also the other type of molecules, synergistic compounds that you want to include into the exosome, okay, to improve their benefits. The way of or the methodology of these custom developments will be exactly the same than in the case of the liposomes. The same time, normally it will take around 6-8 weeks. You know that the first step is to carry out a feasibility study to check if what you want to obtain it's possible or not. Then this 6-8 weeks to produce the first prototypes and carry out the stability test, and then we can send you the first prototype with all the documents to finally send you 1 kilogram of the final product of a product which will meet your requirements. And the final question is, what's the best? Is liposomes or exosomes? Obviously, there is no winner here, okay, because they are not competitors between them, okay. They are quite similar in which related with the structure. They are very similar as vectors. The composition of the membrane is very similar. So probably the difference is in the actives that we are going to encapsulate or the products where we are going to add these liposomes or exosomes. I mean, if you are interested in encapsulation of a pure compound, one pure compound, or not too much, 2, 3 at most, pure compounds with a very high specificity, for example, you want to encapsulate retinol or retinal because you really want to encapsulate the highest amount of of retinol and just retinol to increase the production of collagen into the fibroblast, okay, probably the best system for you is a liposome. However, if you are thinking in a vector which contains also different types of biocompounds, natural biocompounds from plants, which have between them synergistic properties, and the exosome cover a lot of different benefits. I mean, there's a multi-property vector, and for you it's very important to have 100% natural plant-derived exosomes, plant compounds from plants. Probably the best system for you is exosome. And we can also create hybrid system. I mean that we can create exosomes because they are vectors which contains biocompounds, different biocompounds from plants with other compounds from plants, but we can, of course, we can also include synthetic actives if you want. We can include retinol also in the exosome. Of course, the amount of retinol that we are going to encapsulate into the exosome will be slightly lower than in the liposome, because into the exosome, to be an exosome, we have to use a part of the space to encapsulate this type of biocompounds, genetic material that that make a liposome an exosome. Okay, so the room available to encapsulate other actives will be always slightly lower into the exosome than into the liposome. Okay, and but of course, this space that you are using for the encapsulation of these biocompounds, of course, they have their own properties and benefits. So it's very difficult to know in advance which will be the best in regarding the effectiveness. So it's more the type of actives that you want to carry out, and also if you are if you want to have a multi-property vector or a very specific vector. That's all from my part. I hope that you find this webinar interesting, or at least useful for you. And here we have our contact details in case of you need further information or you want to ask us whatever. Thank you very much for your attention, and it was a really pleasure. Bye bye.